Hello everyone. Thank you so much for taking your time to join this webinar. This is Prashant Chauhan, working as a program executive at Bioinformatics Technologies of India. My skills align in the field of bioinformatics and chemoinformatics. I have published articles in SCI Index Journal with good impact factor. So the topic of today's webinar is based on CADD, which is computer-aided drug designing, and the type of computational technique which we use to discover the novel and potent therapeutic drugs. Hello everyone. Thank you so much for taking your time to join this webinar. This is Prashant Chauhan, working as a program executive at Bioinformatics Technologies of India. My skills align in the field of bioinformatics. And the topic which we will cover in this webinar is revolutionizing drug development with computer-aided drug designing toward anti-cancerous molecules. As you can see the word anti-cancerous. So the point which comes in our mind is that why we have selected this cancer disease only despite of having several severe diseases in the world also. So you must have think what makes cancer a major concern. So cancer is one of the most devastating disease across the world and the second leading cause of death with a high mortality rate due to its complexity and inadequate treatments. It has been estimated that approximately 1 million new cases has been found in 2023, that is equivalent to about more than 500,000 cases each day. Although several therapeutic drugs have been implemented for the management of cancer, however, they have severe side effects on the human body. Additionally, many patients with cancer still do not have a cure. Therefore, in this webinar, we will be focusing on phytocompounds that show less toxicity and better efficacy for the management of cancer in comparison to the synthetic drug. The phytocompounds are the plant-derived compound that shows less toxicity in comparison to the standard drugs. Now let's come on the introduction part of CADD. So CADD is a computer-aided drug designing. It is a multidisciplinary field at the intersection of chemistry, biology and computational science. It involves the use of advanced computational techniques and software tools to enhance the drug discovery and development process. CADD also plays a pivotal role in the identification, design and optimization of potential drug candidates. CADD also helps in rational drug design, lead optimization, and the selection of the most promising compound for the further experimental testings. Here is the flowchart of CADD. CADD is of two types. First is structural based drug designing. Second one is ligand based drug designing. Ligand-based drug designing comprises the two parts, which is pharmacophore modeling and quantitative structural activity relationship, which is normally called as QSAR. But in this webinar, we will majorly focus on the structural-based drug designing, which comprises all these mentioned steps: binding site identification, docking and scoring, virtual screening, then compound selection, lead optimization, then the development of novel drug. So here you can see the flowchart of structural based drug discovery, which is a powerful approach in the field of drug discovery that relies on the detailed knowledge of the three-dimensional structure of biological molecules such as protein or nucleic acid to design and develop novel drugs. Structural based drug discovery is rooted in the idea that understanding the precise atomic interaction between a drug molecule and its target protein can lead to the rational design of highly specific and effective therapeutic compounds. So as you can see in this flowchart, there are several steps mentioned to perform the structural based drug designing. The first one is protein isolation and the purification, then the protein structure and elucidation, then identification of target binding site, then the virtual screening of drug-like molecules, and the fifth one is molecular docking and its virtual based screening. The next is molecular dynamic simulation and binding free energy calculation. Then after that you have to go for the chemical synthesis of top hit molecule. Then after the biological evaluation, then the final step comes for the lead molecule and the drug development process. So as I have shown you in the previous slide about the steps to perform structural based drug designing, so the first point is comes in our mind to prepare and download the protein structure. Before delving into the molecular docking, we need to download the target molecule that will be a receptor, and after that the preparation of that molecule become a significant part of the molecular docking to get accurate results. There are several databases available through which we can download our desired protein structure. The PDB Protein Data Bank is the well-known database which widely used for the for downloading the and retrieving the data about the protein structures. Here are the some major steps which we need to keep in our mind before downloading the structure of protein from the PDB. That the protein structure should be of high resolution, that should be of greater than 2 angstrom, and the method of elucidation of that particular protein should be NMR and X-ray diffraction. And we can also go for the organism type on which we are working. Like we are looking for the Homo sapiens, then the organism type of that particular protein should be the Homo sapien. So here is the user interface of protein data bank, where you can search for your desired protein. As I have searched for the 1XM9, which is crucially involved in the progression of non-small cell lung cancer. Here you can see the above mentioned parameter. The method of elucidation is X-ray diffraction, and the resolution is also greater than 2 angstrom. So yeah, we can consider this particular protein for the further studies. And by clicking on this view button, you can visualize the 3D structure of this particular protein also, that I will show you in next slide. So after clicking on that 3D view, you will visualize this kind of structure here, and here you can see the polymer, ligand, and the water molecule also. So before downloading this structure, you have to hide all these ligand and water molecule except that polymer, and then after hiding these molecules you have to download this structure. And when you once click on this ligand, then this red color will show on your screen just to identify the ligand molecule, and when you will click on this water molecule, then the red dot will get appear just to identify the water molecule in between this protein structure. So first we have to hide all these intermolecules and then download the protein structure in PDB format by clicking on the download button given at the top of this interface. So as we have downloaded our protein structure, now the time begins to minimize its energy for further docking process. So either you can use the manual software for the minimization of energy, or you can use the online based server also. So first I will show you how to minimize the energy of that particular protein using UCF Chimera, which is the manual visualization software. So once you click your PDB file in UCF Chimera, then this kind of interface will appear. Then after that you have to click on that tool button. Then after tool button you have to go for that structural editing, then minimize structure. Then after this second window will appear, and finally you have to click on this minimize button. Then after this, the further 3 pop-up will come up, and here are the 3 pop-up which come after that step. So they will ask you about some permissions, about to add the more hydrogen bonds and to add more charges. So you have to just click simply OK, then OK. Then after mark right on this add hydrogen and add charges, you have to click on this OK button. Then your file will get minimized. After minimizing the energy of your protein, you can also visualize how much energy you have minimized through that process. So the option is available in the Chimera itself by simply clicking on that tool button. Then after structure analysis, then after this option atomic calculator, attribute calculator exactly, then this window will come. Here you can analyze the initial energy and the final energy also. As you can see, the initial energy is less than the final energy and it's given in the negative form. So yeah, we can say that our energy is minimized properly. And here is the online server which we can also use for the preparation of your protein, which is known as Yashara. This server performed an energy minimization by simply uploading your protein structure in the given column, and then after you have to type your email address here. You can also enter the PDB ID rather than uploading the PDB file. Then the result will come on your Gmail, which further you can visualize. Till here we have downloaded our target protein and minimized the energy as well. So now the time comes to select and download our ligand for the docking. So there are several databases available through which we can download our ligand, such as PubChem, NPACT, Impact, Zinc database is also there from which you can download your desired ligands. PubChem is the database from which you can select the desired ligand and chemical molecule for the docking purpose. And NPACT is the curated database of plant-derived natural compounds that exhibit anti-cancerous activity. Here you can analyze all the classes of phytocompounds that shows anti-cancerous activities. And after that, Impact is also available which you can use for downloading your ligand structures. So this is the user interface of all those databases: Impact, Impact, and PubChem. In this webinar, we will majorly focus on this Impact database, which is curated for all the anti-cancerous compound. This is the user interface of the NPAC database, where you can see the list of organic compounds which we can select as the ligand for docking purpose. Then once you click on this browse button, then you will gonna see different kind of classes of compound, like such as terpenoid, flavonoid, alkanoid, and etc. And when once you click on any of this class, such as if you click on this flavonoid, then about 329 compounds will get appear on your screen in this manner. Here, according to your interest, you can use any of these molecule, such as epicatechin. I have selected for the docking purpose. And after clicking, you can also see the PubChem ID of this epicatechin here. And when once you click on this epicatechin, you will also analyze the other properties of this particular molecule. And when once you click on this epicatechin, then this user interface will come. Here you can analyze the cancer cell lines also, on which this particular epicatechin have been worked on. And you can also see the conical smiles present here. The use of conical smiles, I will talk about it later. As of now, you can just see where you can download or you can copy the smiles of the epicatechin. I am just taking an example of all this protein and ligand. You can choose any of the ligand according to your interest, and then simply by clicking on this download button, you will get the 2D structure of this epicatechin. And if you need the 3D structure of this particular protein, then you have to copy this PDB ID in the PubChem database, where you will find all the 3D structure, 2D structure, and crystal structure of this particular ligand. So now we will talk about the molecular docking, which is a computational technique used to predict the binding affinity between a protein and the ligand. This technique has become an essential tool in drug discovery and other fields as it allows researchers to screen large libraries of compound and identify potential drug candidates with high accuracy and efficacy. Here you can see this image where you can also see the target and the ligand then equal to molecular docking. So in previous slide I have explained you about how to download and prepare the target molecule and the ligand molecule as well. Now we will perform the docking by using this target and ligand files. So you can perform molecular docking either with the manual docking software such as Autodock, Vina and Pyrex, and as well as from the online docking software such as PatchDock, SwissDock, CBDock2, IgMDock, etc. In this webinar, I will gonna show you about how to perform docking by using CBDock2. CBDock2 is an improved version of CBDock server for protein ligand blind docking, interacting cavity detection, and homologous templates fitting. This will provide you the 3D structure of a protein and the ligand complex. We can predict their binding site and affinity for computational aided drug discovery also. So this is the user interface of CBDoc2. After opening this page, you have to click on this dock button. Then after clicking on that dock button, this type of interface will come where you have to upload your prepared protein file and the ligand file as well. Then after uploading your files in their respective region, you have to click on this auto blind docking. Then you can also visualize the cavity of the protein molecule simply by clicking on the search cavities, or we can directly click on the auto blind docking to obtain docking results. It's totally depends on you and your choice. If you are looking for the cavity sites, then you can also analyze that with the help of the CBDoc2 server. Then when you once clicked on auto blind docking, this interface will pop up where you can see your results of docking by simply clicking on this view result. So here you will see the multiple Vina score on the basis of protein cavity, but usually the first result we will consider because it is the highest negative score, and the more negative the result, the better binding will show, and the stability will also be the high if you will choose the first negative result. You can also visualize the contacted residues in between the protein and the ligand docking binding. By simply clicking on this view button, you can analyze all the residues which are alliance in between the binding in between this complex. CBDoc2 also provide you the several commands through which you can change the orientation and the color of your molecules, and you can also change the structure and the color of your ligand and the protein as well. You can also visualize the interaction, the bonding which has been formed between the complex. Here you can change the orientation according to your interest. You can also visualize the hydrogen bond interaction, ionic bond interaction, pi-pi bond interaction within the complex. Blue color bond shows the hydrogen bonding, green color bond shows the pi-pi interaction, yellow color bond shows the ionic bonding. As I have shown in this video, here you can easily analyze and visualize all the bonding and the residues attached in between the complex. CBDoc2 also provide you the several commands through which you can change the orientation and the color of your molecules, and you can also change the structure and the color of your ligand and the protein as well. You can also visualize the interaction, the bonding which has been formed between the complex. Here you can change the orientation according to your interest. You can also visualize the hydrogen bond interaction, ionic bond interaction, pi-pi bond interaction within the complex. Blue color bond shows the hydrogen bonding, green color bond shows the pi-pi interaction, yellow color bond shows the ionic bonding. As I have shown in this video, Here you can easily analyze and visualize all the bonding. And so this is the final docked result of the molecular docking. Here you can see the proper binding and the interaction of ligand with the protein along with the residues attached to the ligand. So till here we have performed the molecular docking and get our final product, final compound epicatechin. We have to go for the pharmacokinetic and the dynamic to check the toxicity level and the efficacy of that particular compound with our body. So Swiss ADME and the Mol Inspiration are the two online servers which we can use to analyze the pharmacokinetics of the compound. Final pharmacokinetic of the compound shows you the process how the drugs being pursue in your body, such as its absorption, distribution, bioavailability, metabolism, and elimination. As I showed you the previous slide about the downloading of conical smile of that epicatechin. So that conical smile you have to paste over here. This is the user interface of Swiss ADME. Then after copy paste that conical smile, you have to click on this run button. Then after this kind of result you will gonna see where you can analyze the Lipping's rules, the solubility of that compound, GL absorption, BBV permutation, about the molecular weight, TPSA property. You can analyze several properties present over here according to the. It's depend upon disease to disease on which you have working. This is the user interface of Mol Inspiration, the online server which you can also use to predict the pharmacokinetics of your desired compound. Now we will talk about the pharmacodynamic of the compound using Charm GUI. Charm GUI is a user-friendly software tool to develop a molecular dynamic simulation of your particular compound. First, before performing the pharmacodynamics with Charm GUI, you need to register yourself. Then after clicking on the input generator present in the left corner, then if you are looking for the molecular dynamics, click on the solution builder option given at the left side. Then after that, this kind of interface will come on your screen where you have to upload your PDB file, or you can directly use the PDB ID as well. Then clicking on the next button, you can see the protein molecule by simply clicking on the view button. Like you can see that in these two pictures also, and the next button you can press from here also. Performing pharmacokinetic is a bit tedious task, so it will take time to process. Then after clicking on that next step, this kind of screen will appear where you have to select the parameters for the molecular docking. It's depend on you what you will choose for this. Then this interface will come. Here you can view the prepared structure by simply clicking on the view structure. Then this interface will come up. Here you can view the prepared structure by simply clicking on the view structure. Then after clicking on next, this interface will open. Here you have to select the generation option. You can select more than one option and keep the other option by default. The final result which you have to download for further analysis. Here you have to download the .tgz file by clicking on the download button and visualize this tgz file in other software such as NAMD, Ember and Gromag as well. Thank you so much for taking your time to join this webinar and listening me carefully, and stay tuned for the workshop on the same which will be organized in next month. You can kindly register for that also. Thank you so much.